The landmark discovery of the double helix structure of DNA 50 years ago marked one of the most significant scientific achievements in human history.
Identifying the double helix by Watson and Crick 50 years ago was one of the top, probably two scientific achievements of the last probably 400 or 500 years, because it allowed us to identify what could be the single most important compound in biology.
This discovery enabled scientists to explore the basic building blocks of DNA. Coinciding with the 50th anniversary celebration of DNA, last month also marked the completion of the Human Genome Project, a collaboration of over 1,000 scientists from 6 different countries around the world.
The Human Genome Project is basically an endeavor to sequence or to determine the nucleotide sequence for every chromosome in the human body.
Since the beginning of human history, people have wondered how human traits, from eye color to hair texture, are inherited from one generation to another. The DNA molecule is considered the single most important molecule in all living things. It is the biochemical instruction book that regulates the formation of every single cell and coordinates all functions of every cell, molecule, tissue, and organ in all living organisms. The human body is made up of billions of cells, and inside the nucleus, at the center of each cell, lies a set of 23 paired chromosomes, each shaped differently. Collectively, these chromosomes make up the human genome. Each chromosome, in turn, embodies our DNA. The DNA molecule is shaped like a twisted ladder. Its bases are made up of chemical bonds, namely adenine, thymine, cytosine, and guanine, or the letters ATCG for short. These chemical bonds, called nucleotides, line up in pairs to form one long sentence along the DNA molecule. Our genes, moreover, can be thought of as distinct words or segments along this sentence. What scientists have managed to figure out is the order and position of all these base pairs for each of the 23 paired chromosomes that lie within the human cell. With this final piece in place, scientists have now confirmed that all humans share the same DNA.
The human genome is made up of approximately 3 billion nucleotides. And the claim is that we're 99.9% similar. So that leaves about 1 million nucleotides that are different between various populations or groups of people, or between two individuals. And it's those million differences that make us short, tall, light skin, dark skin, blue eyes, brown eyes, long arms, short arms, big feet, small feet. And it's those million differences that make us who we are. This is the genome era. We now have the genome in our hands. Now it's time to take that data forward to see if we can use it, again, for me, most importantly, to answer important questions in health.
Dr. John Carpton is head of the Cancer Genomics Division at the National Human Genome Research Institute, located at the National Institutes of Health in Bethesda, Maryland. His research is focused on finding the genes that trigger prostate cancer.
So I think the Human Genome Project has been absolutely critical to my work, because it allowed us to have access to all the sequence of all of the genes that we needed to screen in these patients and their unaffected family members. And having access to all of the genes in the human genome is very important, because you don't want to miss any genes. You don't want to only look at half the gene where the gene that's causing it may not be in your experiments.
With the full sequence of human DNA now clearly mapped out, scientists can now begin to identify, prevent, and diagnose all sorts of diseases, from prostate cancer to Alzheimer's disease. This new map of genetic information, however, presents scientists with ethical and legal concerns, such as who will have access over people's genetic information and how will it be used. Vince Bonham is senior consultant to the Director on Health Disparities at the National Human Genome Research Institute.
Our genetic information can be used for a variety of different reasons. Some of them are clearly going to improve our health, but potentially others that could be used to harm us through discrimination, as well as could be used by law enforcement agencies and a variety of other organizations that could have access to that information. One of the things that's clearly a focus of Congress is, can we develop a comprehensive genetic discrimination law that would protect individuals across the country?
These laws would prohibit healthcare insurance companies from declining coverage to certain patients because of their genetic history or predisposition to certain diseases. Bonham is also concerned about how genetic information will be used to define race.
Clearly, race matters in our country, in our society. It is a social construct and a political construct that has such a history in our society. I think there's going to be a number of challenges that we have. I wouldn't call them necessarily drawbacks. I think there's going to be challenges related to the issue of how we understand race in genetics, which I think is going to be a very important one, particularly for communities of color, and how scientists use that information to identify differences in individuals.
While these issues still need to be addressed, Bonham remains optimistic about his work ahead.
We are on a dawn of a new era. The genome era is just beginning. We are not in post-genome era. This is the genome era. And our challenge is to make sure that it benefits everyone, that it doesn't just benefit the few, the rich, but it benefits us all. And I think that we have a great opportunity to do that.
I'm Dr. Aziza Bakouche reporting for Evening Exchange.